Hello, my name is Mr. Anderson, and today we're going to talk about a hybrid cross. Hybrid crosses are how we take uh, heterozygous individuals or homozygous individuals and cross them to see what possible outcomes you can get for the offspring. Today we're going to take two heterozygous individuals for the trait of smooth or wrinkled pea pods. This is part of the experiment that Gregor Mendel did uh, to actually discover all this. He took pea pods or peas and crossed them or self-pollinated them and was able to find out that they were able to produce offspring in specific ratios. So a heterozygous is just an individual that has uh, one dominant, this capital S, and one recessive, this lowercase s, in their possible offspring. So what happens is you set up a nice little cross here, basically a tic-tac-toe pattern right here, and you take one of those s's and put it here, and you take the small s and you put it down here, and then you'll take another individual, big S, little s, and do the same thing. Put the big S here and the small s here. So let me get rid of those lines because they'll be kind of annoying otherwise. So we take one of the big S here, small s here, big S here, small s here. And then what we will want to do is we take, move this over slightly, we take this individual, this S, big S, and put it here and here. Basically any of these rows that they can be in, you put them. The little s, same way, it goes here and here. And on the cross section, you take this big S and it goes to this section, and the far one, the little s, to here and to here, which would give us a big S here, big S, little s there, big S, little s there, and two small s's here. So you can see that we have this big S carrying down to here and here, and this big S carrying on to here and here, this little s carrying to here and here, and this little s carrying to here and here. What this gives us are two different ratios. We want to figure out what the genotypic ratio is and what the phenotypic ratio is. So the genotypic ratio is the ratio of homozygous dominant, heterozygous, and homozygous recessive. So for the homozygous dominant, we're looking for any individuals that have both S's. This one right here with two positive S's is a homozygous dominant. So we want to go ahead and put that in here as 1. Homozygous recessive is the opposite. It is two small ones, which we have one of right here. So let's go ahead and put that in there. Now heterozygous are the same phenotype that we had to begin with. This big S, little s. We have two of them, right here and right here. So let's go ahead and put them in, which gives us 2, which means our genotypic ratio is 1 to 2 to 1, which is the ratio of dominant, homozygous dominant to heterozygous to homozygous recessive. The next thing we want to look at is the phenotypic ratio, which is the ratio of which trait shows. So looking at this, we have 2 different possibilities. You can have a smooth and you can have a wrinkled. Since we know that wrinkled only shows up when you have recessive, we can see right here there's only one possible wrinkled seed right here. So that would give us 1 for the wrinkled, and all the other possibilities are all smooth, so that would give us a 3 to 1. So you have 3 smooth to 1 wrinkled, which means our phenotypic ratio, the ratio of what traits show up, would be 3 to 1. Well, that's all I have time for today. The next lesson we're going to go over a dihybrid cross, which is the same basic concept here, except we're going to look at 2 traits instead of 1. See you next time.